You're at the cutting edge of cardiovascular sciences. I'm your host, John Cooke. I'm professor and chair of the Department of Cardiovascular Sciences here at Houston Methodist in the Texas Medical Center. And we have a very special guest for you today. Dr. Li Lai is one of my faculty members in the Department of Cardiovascular Sciences. She's going to introduce our guest.
Good morning, everyone. Today is my privilege to introduce our special guest, Dr. Ali J. Marian, who holds James T. Wallison Distinguished Chair in Cardiovascular Research, Professor of Molecular Medicine, Professor of Internal Medicine,
James T. Wallace Distinguished Chair.
I am
Director of the Center for Cardiovascular Genetic Research at the Brown Foundation Institute of Molecular Medicine at UT Health Center in Houston. Dr. Marian obtained his MD degree from Tehran University in Iran, and then he completed an internal medicine residency in Chicago and a cardiology fellowship at Baylor College of Medicine. He also completed an American Heart Association Buger Foundation Research Fellowship in Human Molecular Genetics at Baylor College of Medicine. Upon completion of his training, he was appointed as faculty at a section of cardiology at Baylor College of Medicine. He then joined the Brown Foundation Institute of Molecular Medicine as professor to lead the Center for Cardiovascular Genetics since 2006. Dr. Marian is recognized for his research achievement in human molecular genetics and the clinical aspect of hereditary cardiomyopathies. And his clinical relevant studies have garnered him a lot of NIH fundings, as well as numerous awards, including Established Investigator Award from HA, the Lifetime Achievement Award from the International Academy of Cardiovascular Sciences, among others. He's also currently served as editors in high-tier journals, including Circulation Research, Journal of Clinical Investigation, etc. So today we're very excited to have Dr. Marian in the studio with us, and we're looking forward to hear about your insights in cardiomyopathy.
AJ, it's a pleasure to have you on the show.
Thank you, sir. Thank you, sir. It's a delight to be here. Thank you, Dr. Lai, for your very kind introduction. John, you've always been a pioneer in doing innovative ideas, and I'm grateful to you for the opportunity to be here for this very interesting presentation that you have, which is probably one of the unique things that in the country that you have this program, that you have people to come here and chat, as well as talk about their scientific discoveries. Thank you, John.
Sure, AJ. Well, you have a remarkable seminar series yourself, the Willerson Seminar. Tell us a little bit about that, about your own educational activities.
I think, in this day and age, that really science is moving very fast. It's exceedingly important for us to have this type of programs, whether it is in person or virtual, to really get together and discuss, critique, analyze the existing data in order to gather insight into the future research programs. I think these educational activities are exceedingly important for the young generation, as well as for the geriatric ones. Indeed, really, all of us would benefit from more critical, in-depth, comprehensive analysis of the existing data. It is the journal clubs, seminars, webinars are exceedingly important part of our educational aspect, and I really advocate for it. And we were very fortunate to do that James T. Wallison MD Cardiovascular Seminar Series before COVID, and after COVID now, Baylor College of Medicine has taken the leadership on that and runs this weekly seminar series. In addition to that, we also did run a webinar series through a journal that we were editing, which was also, I thought, was a very valuable thing to do for all in the cardiovascular scientific community. So I'm very much for this. I'm not a classroom teacher type of guy, and I'm very much into interactive, analytical, critical analysis of the data, which I think is exceedingly valuable.
You know, I've really enjoyed your own seminar series across the street, Texas Heart Institute. But I remember it was about, I don't know, maybe 5-10 years ago, you joined us here at Houston Methodist and gave us a grand rounds that I found fascinating about the fortunate generation.
Yes,
Why are we the fortunate generation?
Yes. John, for those of us like you and I who have been around and have looked back to the history of how we train and what tools and interventions were available to take care of our patients, and compare it today, what is available, indeed we are a fortunate generation. When I grew up in medicine back in 1970s, we had only digoxin and Lasix for treatment of all cardiovascular diseases. And then of course gradually aspirin, physician health study came, and then after that, beta blockers came with BHAD study, and anticoagulation came for myocardial infarction, and so forth. And look at the modern generation now. Now we're dealing with SGLT2 inhibitors and GLP-1 receptor agonists, a large number of monoclonal antibodies in other fields of medicine, which are used practically to target some of the cancer patients to the point of eliminating the cancer. We indeed witnessing this evolution from 1970s to today, although I'm not that old, but it is indeed mesmerizing to see how much advances have been made. Look at the lipid disorders. It's just amazing what genetics and genomics have made impact, and so many RNA-based therapeutics are coming on board that really changing the practice of medicine. In my own field of focus, hypertrophic cardiomyopathy, the pioneering work of Seidman's, John Seidman and Cooke at Seidman, discovering the first causal mutation really had watershed effect and led to figuring out the mechanism by which actin and myosin interact, and development of targeted therapies that specifically target this underpinning mechanism for treatment of cardiomyopathies. It's a fascinating world. Really, this is Huxley's Brave New World in a different, in reality, really, in a different format, but in reality, we have been very fortunate to witness all the changes.
You told us at that seminar that we're the fortunate generation in part because of the Human Genome Project and what we learned in terms of the variation of the genetic diversity of our population can give us some clues as to what we can do for cardiovascular disease. And tell us a little bit about that.
Your point is absolutely well taken, John. I was, of course, scientifically just a neophyte, and training in cardiovascular genetics and human molecular genetic program was just emerging. Human Genome Project was a massive project. I believe it was $13.2 billion, if I'm not mistaken. And of course, there was this crazy guy, Craig Venter, and he thought that all NIH investigators were a bunch of loonies, and he went out on his own and do shotgun cloning and practically beat the NIH and Human Genome Project. Nonetheless, together they came together on, it was June 5th or 6th, 2001, with Bill Clinton was the president, and they announced that indeed they have sequenced Eric Lander, Craig Venter, Bill Clinton on TV announced that we have sequenced the Human Genome Project. Many critiques at that time, they said this is massive amount of money spent for a descriptive project to show what is the alphabet of Human Genome Project. What is the significance of this? What is the impact of it? Today, basically all modern medicine derives and originates from human molecular genetics, Human Genome Project. And knowing the genome sequencing has enabled to do all the GWAS studies to find the susceptibility allele to various traits. For monogenic diseases, practically to a large degree, they have been deciphered. The molecular genetic basis, a significant part of classic monogenic disease, although a subset of them are oligogenic and polygenic, still needs to be deciphered. But that really had changed the practice of medicine. A lot of it, lipid disorder is the best example. I mean, look at every day there is a new siRNA, RNAi-based therapeutic targeting. The other couple of weeks ago, I was mentioning there was this therapeutic targeting using CRISPR-Cas for genes that produces too much proteins or abnormal proteins that aggregate, such as amyloidosis, and deposit in the heart and other organs and causes. Those could be easily targeted by CRISPR's Cas. They are the best candidate, probably, those type of genetic disorders to target with CRISPR-Cas technique. So the impact of Human Genome Project could never be underestimated.
You know, I remember one of the most striking things about the Human Genome Project discoveries was that we only have 20,000 genes, right? 20,000 protein coding genes, and everyone was astonished because, you know, simple unicellular organisms have more genes.
That's right.
How could that be?
Rice has the biggest genome.
Rice has the biggest genome.
Yeah.
How could that be?
Yeah.
And you know, now we know a lot more about
the
non-coding
part. Early on, they were estimating there are about 100,000 protein coding genes, partly because the data was originating from isoforms of the proteins and RNAs included to consider them as independent genes. Then we realized there are about 20,000 large protein coding genes, but now there's also the new information that there may be much more peptide coding genes, that, you know, small peptides are getting coded from the unknown so-called part of the genome, because genome is pervasively transcribed and likely pervasively translated. Anyway, translation machinery finds an RNA could translate, but the proteins, the vast majority of the translated protein, probably are unstable and decay. But some of them will make it.
Yeah.
So we will have, look, angiotensin 2 is an 8 amino acid peptide. Right? I mean, so it would not be surprising there are small proteins that are comprised of 3, 4, 5 peptides and coded. So the number, you may argue, may be a little bit more than 20,000 or whatever it is, but nonetheless, it is intriguing. Or on different aspects of it. I mean, look at non-coding RNAs that come from rest of the genome.
Exactly, right.
It is, so impact of the genome is the fundamental. Genome is the stage where our orchestra plays. The rest are instruments. How you put the chairs, how you put the instruments, you get different music, which is the phenotype that we see. Exactly. The underpinning mechanism of our being is our genome.
Right. And all of these non-coding RNAs are regulating the orchestra, as you put it. Those 20,000 genes can be markedly regulated by everything else. What used to be called junk DNA, because we didn't understand what it did, that 98% of the DNA that doesn't have protein coding capacity has other effects to regulate the genome. Yeah.
No question. You are absolutely on the mark. Indeed, I think biology is an orchestrated chaos. I mean, these molecules are constantly in their Brownian motion based on atomic interaction between components of the proteins. You know, this C likes that little N there, they attach together. This oxygen likes that little phosphate attached. The whole biology is molecular interaction. And where they are located spatially, at what moment, forms the phenotype. So I'm not surprised there is enormous complex interaction. The vast majority of modest effect sizes, which we are incapable of detecting. And those interactions that are strong affinity, they form strong bonds. They are the one we see that leads to gene transcription, for example. Even transcription factors, they don't go just sit on the promoter, it says I'm going. They're just constantly searching the genome, little attach here, off, attach there, off, until they find atomic interactions that are stronger between the molecule. And that leads to transcription of RNA. And those transcriptions are also not continuous, permanent. They are, cyclic. Often. They are burst-like. They come and go until the full transcription are generated through probably a lot of special fragments binding together, making the form. It's a fascinating complex. I'm not sure we will ever get to the end of this journey, because as we get there, some new players come.
Absolutely.
It's a fascinating field.
Makes it fun.
Yeah.
Well, on that note, maybe we should go ahead and get
Absolutely.
started on your presentation, because it's a
I
nice
will
segue.
be delighted.
Nice segue into disruption of the genome.
Yes,
which
indeed.
you're going to talk about.
So today what I would like to present to you is something that we serendipitously stumbled upon. My area of research is predominantly on genetics, genomics, and pathogenesis of hereditary cardiomyopathies, which are primary disorders of heart muscle cell, cardiac myocytes. Of course, the phenotype is a complex, intertwined interaction, often nonlinear and often random, stochastic, between numerous constituents of the myocardium. Although cardiomyopathies are primary disorders of cardiac myocytes, the interaction of these with fibroblasts, endothelial cells, and other cells in the heart leads to ensuing phenotype that we clinicians call it cardiomyopathy. The primary stimulus is in the gene mutation. That affects primarily cardiac myocytes. The mutant cardiac myocytes interact with other cells, lead to cardiomyopathy. So the phenotype is complex multicellular. The origin is a mutation in cardiac myocytes. So that's my area of expertise, and not expertise, forgive me, area of research and interest. I still don't know much about the disease. And so working on this, we stumbled on evidence of activation of double-stranded DNA break in cardiomyopathies, which was the topic of my presentation today. And I'd like to make this, although I'll present in the context of cardiomyopathy, make a point that this is really, I'm going to be a little silly and say applies to everything. We can discuss this at the end, how. But I will present largely in the context of cardiomyopathy, but I believe it is an essential component of the genome, imprinted our genome to develop double-stranded DNA break, and consequently imprinted of genome to age, imprinted of genome to have genomic instability, and so forth. That's the reason I think this field has a much broader implication, which I'm really is not, an expert in this area at all, by no means, but we are entering the field. Is a journey, which I will conclude that this is an ending journey from my point of view. So let me start. We were talking about Human Genome Project. This is the beautiful art of nature. This beautiful crystal you see is a fascinating molecule. It is the molecule that won the Nobel Prize for Arthur Kornberg. This fascinating molecule is DNA polymerase. It has been assigned to replicate every single nucleotide in the human genome, of which we have 6.4 billion units, to replicate it in all of our human body, in all of our cells, of which we have 37, 10 times to the 12, I don't know what that number is, and replicate that with an exceedingly high fidelity. This is an amazing molecule. This molecule is an error rate of approximately 1.2 error for every 100 million units of DNA that puts together. Imagine if you had a scientist who had that accuracy. Imagine if you had a postdoc who could do the experiment with that kind of fidelity. One error every 100 million units. But that error is the essence of life. If we did not have that error, we would not survive. We would be eugenic, and COVID will come and destroy us all. That error is what we are, how we look different, how we think differently, is the color of our skin, content of our mind, and susceptibility to disease, and how we respond to treatment. That's the occasional error of that molecule of nature. That molecule of nature does occasional error, and those errors that are rare are the key to understanding biology, the key to understanding life. Johann Wolfgang van Goethe has this beautiful statement that it is in the anomalies that nature reveals a secret. It is so true. It is so true. So I always in my career from the very beginning, because I grew up in the era of monogenic disease, genetic discoveries such as cardiomyopathies, lung QT syndromes, and others. So I always go for rare errors. Why? Rare errors, their effect size is large. Common errors are ancestral. Their effect sizes are small, and they survive the evolution. Therefore, they can have a massive error, the common genetic variants. It is the rare errors that gives us insight. I will use one example of that rare error to gain some insight into pathogenesis of one form of human disease. And that rare error is this. It is truly fascinating to see that tiny error. Look at those two amino acids. Aspartic acid, asparagine. They're largely identical, except that one hydroxyl group had changed to an amino group. Subtle change in a gene called lamin A. This subtle change leads to this kind of phenotype. This is the beginning of our endless journey. A young lady presented to this hospital, Houston Methodist Hospital, years ago, came with acute myocardial infarction at age 29. We looked to see, do you have diabetes? Do you have familial hypercholesterolemia? Do you smoke? I mean, what gives you heart attack at age 29? We couldn't figure out. She had massive coronary artery disease, underwent coronary bypass surgery, multiple PCIs, comes back within a year or so, develops valvular regurgitation, valvular insufficiency, undergoes aortic valve replacement, mitral valve replacement in her 30s, and then within a few years developed massive conduction defect, atrial fibrillation, requiring pacemaker, develops heart failure, mostly a restrictive type of cardiomyopathy, and dies at age 41 while waiting for heart transplant. We look at this picture, say, my God, she has all diseases that elderly gets. This is a progeroid syndrome. If we look around, we were not very knowledgeable at that time to look for other signs of progeria in this case. She had a little bit of tight skin. Whether she had dermopathy of progeroid syndrome, I can't tell. But we called it, this is a non-syndromic form of cardiac aging, non-syndromic cardiac progeria. So we embarked to figuring out the genetic basis of it, which led to discovery of that rare anomaly that I mentioned to you. And then we moved on to gain some insight. Of course, we did not have hearts with that rare mutation, because impossible, because this was the first in the world. Subsequently has been validated by others, identified this mutation causing progeroid syndrome. But we had 5 hearts with pathogenic variant in the lamin, truncation variant.
Now, can we pause here just for a moment, AJ, and talk about what is lamin A? Where is it found in the cell? What is its function? Maybe you're going to get to that.
Yes,
let's
yes,
pause for a moment.
I will come to that after this introductory slides, and we discuss about lamin, how it potentially causes these things as a nuclear envelope protein. So we took those 5 hearts and we did RNA-seq to gain some insight about biological pathways that are dysregulated. One of them that showed up was T53, shown in red on that slide. T53 was quite remarkable, and we did validate this by multiple other methods, not only at protein level as shown on the Western blot, but also T53 target genes were activated. So T53 was activated in the heart of this lamin cardiomyopathy patients. So what is a T53? T53 best known in the cancer field. Let's advance the slide. And it is, of course, T53 is called the guardian of the genome. And because when T53 mutates, you get lung cancer, you get colon cancer, you get breast cancer. Indeed, half of the breast cancer has T53 gene, half of the lung cancer has T53 gene. Okay, now we have advanced, let's advance this slide.
Let's go forward.
Let's,
Henry, go forward. Perfect.
Forward.
One more. Again.
This is backwards.
Okay, now we can move past this slide. We said that T53 is a DNA damage response gene. It is the key component of guardian of the genome, basically. It's not the only one, it's a key component of it. So what is DNA damage? Our DNA are constantly, incessantly exposed to internal and external obnoxious agents. External is obvious. X-ray radiation, ultraviolet light, nuclear bombs are the best examples where they damage the DNA, destroy the numerous type of internal stressors of DNA, which is the topic of DNA damage in cardiomyopathy, is metabolism, production of reactive oxygen species, alkali adducts that bind to DNA, proteins that are generated that go and bind to DNA cause double-stranded cross-linking. These are every day produced in our genome. By some estimate, every mammalian cell gets between 50 to 100,000 DNA lesions every day, every cell.
Wow.
And a majority of them are benign, relatively. Their effect is small. Oxidation is one of the most common. Guanine constantly get oxidated. And that oxidation easily could be detectable with various methods in cardiomyopathies, which we have detected. And they are listed on that slide here. And this is, I'm not sure, is this the pointer?
Yeah.
Okay. So, so I went back. Let me see. We're going. This goes. Let me go. Okay. Let me go back to that. Okay. So, the number of internal stressors damage the DNA. The most serious ones are cross-linking. Cross-linking is the most damaging one. An example of cross-linking is chemotherapeutic agents.
Yeah.
All these, cancer chemotherapy agents, they bind to DNA. Adriamycin toxicity is the classic. And those, when they bind to DNA, affect the normal function of DNA, which we will come to that, which is mainly replication, transcription. So the DNA damage pathway, this is my simple classification, could be classified into 3 groups. I will not go with the order listed in there. Anytime there is a damage to DNA, the first thing cell does is says, shut down the engines. And the way it shuts down, activate checkpoint pathway inhibitors. And that is ATM, ATR, and T53 and cell cycle regulators to stop cell functioning because of defective cells. They want to cause cell death and destroy the cell. That's the first thing when there's damage to DNA, cell does. And then it says, workers, hurry up to the damaged site, repair it. The DNA damage pathway. There are about 5-6 different pathways that are activated in response to DNA damage. And some of them are cell cycle dependent, some of them are independent of cell cycle. And the third thing happens when there's DNA damage, some of this damaged DNA leak from the nucleus in the case of nuclear DNA, or from mitochondria, in the case of mitochondria, to the cytosol. When it comes to cytosol, evolution has given cells ability to defend to external and internal DNA. There should be no DNA in the cytoplasm because cell now has equipped with this proteins that anytime COVID, anytime any virus invade the cells, injects its DNA, RNA into the cytoplasm, cell activates DNA's RNA sensing proteins to destroy that cell. And that classic one of them, there are about a dozen of them, cGAS is the most famous one, for which this year they gave the Vascular award to Dr. Chen from UT Southwestern. And cGAS activates a cascade event that leads to what John called inflammation. Induces inflammation in the cell and leads to aging plus other phenotypes that we will discuss. So this is a DNA damage pathway that we think is activated in cardiomyopathy. To test that, we did a number of, I will go through some of these data slides relatively fast. The first one you see in the green is a relatively simple assay. It's based on the fact that DNA is negatively charged molecule. You put DNA nucleus in a gel, agar gel, and electrophoresis, it will migrate to the positive end. And that's what happens, and gives a picture of comet light that you see on that slide there. Comet is called comet assay. In the lower panel, you see the comets. And then we did test bunch of, this is human heart, by the way. We checked them in a bunch of molecules that are activated in response to DNA damage, including H2AFX, which is a sensor of DNA damage. They are activated in human heart with lambing cardiomyopathy. Then we went on to identify these cytosolic DNAs, the damaged DNA. In this slide, you will see this is H2AFX. There are three different genotypes. The first two genotypes are control, the first two panels. The third panel is a mouse model which we have of cardiomyopathy, which we have isolated myocytes from them and stained the DNA with cyber green and then DNA damage marker H2AFX, which is an atypical histone phosphorylated form of it, with red. You see that in the middle of two nuclei, you see a green dot. The arrow is displaced a little bit, and that shows when the format of the slide changed, the arrow has displaced. You see that really is a piece of DNA that outside the nucleus and is stained both for green as well as with DNA damage marker H2AFX, and on the overlay images you can see it in the cytosol. So this is a cytosolic DNA. DNA is not supposed to be in the cytosol. This nuclear DNA that has exited the myocyte in the cytosol. We have done the same for mitochondria. I will go through the next three slides. This is another DNA marker, and this one is T53 binding protein 1. Also, you can appreciate the DNA outside the nucleus. Look at the middle. In the third row, which is our cardiomyopathy, the other two are controls. You can appreciate the arrow. And then this is the third DNA marker, which is a marker active transcription, because we have the hypothesis that these damaged DNA are released at the site of active transcription. So that is a K4 acetylation. Again, you can appreciate green is. Look at the lower panels, which are the magnified one, which is from a mouse model of cardiomyopathy. You can accept these are DNA in the cytosol and is acetylated. So, and then we have done this also by qPCR, measuring nuclear DNA as well as mitochondrial DNA in the cytosol, separating the cytosol from other components. Again, we see in about half of the cells that we examine, half of the heart that we examine, we can detect cytosolic DNA. So then, of course, now the question is, you detected cytosolic DNA. If you look at the panel to your left, to the right of the screen, and you can appreciate that's a cytosolic DNA sensing protein pathway, cGAS, STING1, TBK, NF-kappa B, and IRF3. the lower gene set enrichment analysis curve shows that the target of IRF3 are activated, and the middle one is the Western blot, a number of proteins that are involved in cytosolic DNA sensing pathway. Without going to a specific one, they are activated, and the panel to your right is the quantitative data. The cytosolic DNA sensing protein pathway is activated in human heart with lamin cardiomyopathy. Let's move the slide forward. Okay. So this is in the human heart, and I would not go into detail of this. We have analyzed this in 6 different mouse models of cardiomyopathy, and all 6 we do find evidence of activation of both nuclear component of DNA damage pathway, which is T53, ATM, checkpoint pathway, as well as cytosolic DNA sensing protein pathway in all 6. So we think DNA damage, activation of DNA damage pathway is a common event. And I would not be surprised, and indeed I think it is a common process of aging, and all of us have some degree of activation of DNA damage as we age. So with that, then we embark to figuring out, then I will come to molecular mechanism of lamin, to embark to identify, okay, these are markers of DNA damage, double-stranded DNA break. These are protein markers of double-stranded DNA break. Where are these breaks in the genome? Mm-hmm. So we use the novel technique, which is referred to as end sequencing. I will not go through technical aspects of it. Suffice it to say, you have to process the whole thing in agar gel to prevent external DNA damage after extraction, because DNA breaks after you extract it. And you do that, and you label the ends with biotinylated probe through this process, and ultimately you pull it down with streptavidin and do short read sequencing. And each peak in the reds are from mouse model, and the blacks are from control. Each peak is one site of DNA damage, and it's shown on your right is the nucleotide sequence. The top panel are the wild-type mouse, and the lower ones are the lamin cardiomyocytes, and you can precisely identify the nucleotide in which double-stranded DNA break has happened. Of course, when we submit this for publication, reviewers said, so what? In science, you never question so what. You say so what, which means I cannot imagine. The same that Galileo walks and said, I found the earth is not flat, is spherical. And the antagonist says, so what? Just to make it light. All right. Oops, I went too far. Forgive me. The next question is, where are these double-stranded DNA breaks? And these are electrophotogram of the sites of the DNA break genome-wide. They are scattered all over the genome, globally speaking. Then we have to this is 30,000 feet view, then you have to go pinpoint. So we compare, this is the fascinating part, we find basically one double-stranded break in every 100 cardiac myocytes. Mm-hmm. And these are only 3 weeks old cardiac myocytes. So double-stranded DNA break happens very early, and I will tell you why. And then we compare the lamin myocytes, lamin deficient myocyte to wild type, we identified 6,774 differential double-stranded DNA break, which it is the fascinating part. Differential double-stranded break at 0.01 signal, false discovery rate. Interesting part of it is, of these 6,774, only 8 were in wild-type differential. Mm-hmm. 6,700, I forgot the number, my math. 68 of them were unique to lamin. Mm-hmm. It is fascinating which means comparing lamin. Sure, wild type happens because these double-stranded DNA breaks are very dynamic. I'll come to that. But it's intriguing that majority of it also speaks to fidelity of the system of detecting this double-stranded DNA break that we did. So the first question is, where are they located in genome? Vast majority of them are whether in the wild type or lamin deficient or differential. They are located at the site of transcription start site, where the gene transcription happens.
Right.
This is the first evidence that we learned that double-stranded DNA break may be in non-replicating cells, such as cardiac myocyte, may be related to transcription.
Okay. Let me just pause here for a moment.
Sure.
So as I understand this now, in the lamin deficient animals and in your lamin mutant patients, you're seeing increased evidence for DNA damage and DNA double-strand breaks. And furthermore, these double-strand breaks appear to be occurring at transcription start sites. So these are the sites where transcription of DNA to RNA is occurring. Did I get that?
That's perfect.
Okay.
Nice summary, John. Maybe you should give the talk. You do better than I do. So that's the first evidence. The next we looked at genome. Of course, it's fascinating. Every time there is a major discovery comes, a lot of other things get subsided, suppressed. Genome is, of course, Watson-Crick model is right-handed double-stranded DNA helix, and which is called the one in the middle, bDNA. There are a lot of other structures of DNA. There are DNA that goes the opposite way. There are DNA that are totally left-sided, and that's a zDNA, and so forth. We look to see whether double-stranded DNA breaks are more in atypical component of the genome. We did not see a difference. Whether it is, again, that may be technical, but we do not see differences in localization of double-stranded DNA to non-bDNA structures. The next question, which fascinates me, is genome also has very fascinating type of structures. 4 guanine come together, bind together through an ion, and then forms quadruplex. That's called G quadruplex complexes. And bioinformatically, one predicts there are about several 100,000. It's more sequencing data suggests, again, incomplete, there are probably about 700,000 of these structures in the genome. But they are small structures. The minimum is 4. Probably average size is somewhere around 30, we're guessing. There's no accurate number. So a small fraction of the genome, less than 1% probably, are G-quad structure. If you map the DSPs to those, it seems to be 40% of the sites that have DSP have G-quad structure propensity. So one positive says, our G-quads are susceptible to double-stranded DNA break? I suspect they are. And that's one of the focus of our research, because G-quad are susceptible to oxidation. And if they are susceptible to oxidation, they are predominantly located at promoter region of the genes. Remember, it was also mapped to TSS. It brings it together. And that's where the DNA damage mostly happens, at the site of the G-quads, where the structure is unable to, transcription machinery unable to proceed, which I will elucidate shortly. Now it brings the John question. So you showed a lot of descriptive data, Dr. Marian. Tell us how this lamin causes this problem. Lamin is a fascinating protein. Shown on the left side of the slide is a myocyte. Red is the nucleus. Red is the lamin, blue is the DNA. And you can appreciate lamin encompasses the DNA and interacts with DNA in the cartoon, that the artistic rendition of your humble speaker here, and interacts with the genome through domains that are referred to as lamin-associated domains. And I will tell you briefly about lamin-associated domains. And we embarked to identifying lamin-associated domains in the human heart. So we tagged human heart with a protein which is known to largely, not exclusively, stain cardiac myocyte, and that PCNA. And PCM1. we stained the myocytes with PCM1 and fact sorted those nuclei, isolated, and performed chip sequencing to identify, this is the fact sorting of those, I'll go through that, to identify lamin-associated domains. On that panel to your left, the top three in blue are the wild type. The bottom three are the human lamin mutation cardiac myocytes. This is called a lamin-associated domain. Both wild type and lamin myocytes have signal. And this is a constitutive LAD, is a LAD that is present in wild type as well as is lamin deficient. About in the human heart, we identified about 300 LADs, approximately each of about 2 million base pair long. Of those, about 2/3 or 1/3 of them are constitutive LAD that are shared in both normal human heart as well as lamin deficient. There are conditions like this. The first one is a LAD that is present in the control heart but is absent in lamin heart. The lamin has lost ability to bind to chromatin in this fragment, in this region. The next one is the normal heart doesn't have a LAD. The lamin heart has a LAD. This is the region where lamin now binds to chromatin that normally doesn't bind. This is referred to as facultative LADs. LADs are also dynamic. They are not fixed and stable. Maybe about 1/3 of them have a tendency to bind to genome and hang around, but a lot of the others are dynamic. They get redistributed, they gain and loss. Here is an example of it.
Let me ask you a question about, maybe you're going to talk about it now. What's happening to gene expression in lamin-associated domains?
Absolutely. Thank you, sir. We come to it in the next 2 slides.
Okay, thank you.
So here is an effect of LADs on DNA methylation. First, look at the control. You can the intensity, green is the lowest, red is the hottest. You can appreciate both in control as well as in lamin cardiomyopathy, LADs have more DNA methylation than the control heart. And in the case of comparing DCM to control, LADs in DCM have more methylation than control. So one of the characteristics of lamin is indeed to recruit DNA methyltransferases and methylate the DNA. And then the other happens to gene expression. Here is when you have LAD binds to chromatin. Blue means less gene expression on those heat maps. Red means more gene expression. In DCM, gain of LAD is associated with suppression of gene expression. When you lose LAD, when lamin doesn't bind to chromatin, gene expression goes up. So the lower panel is an example of one genomic region. To your left is a site of LAD, gain of LAD in this case, because we're comparing wild type to DCM, control to DCM, and you can see each dot is one transcript. There are few dots there compared to loss of LAD, which is the panel to your right. You see more transcript there. So LAD methylates DNA and suppresses gene expression. And we move, because now we wanted to do mechanistic studies. In human heart, we restricted the samples available. We moved to mouse myocytes and also defined LAD in mouse myocytes. And by and large, we see the similar finding. About 1/3 of them are constitutive LADs, about 2/3 of them are facultative LAD. And we do see that both in human as well as mouse cardiac myocytes, about 20-25% of the genome or chromatin binds to LADs, binds to lamin. They are LADs. LAD comprises about 20-25% of the genome. And here is effect of LAD on gene expression. And the so-called violin plot shows that when LAD binds to chromatin, gene expression goes down 16-fold. And the middle panel is, again, each transcript in the genomic region is in the LAD and non-LAD region shown. Again, you can appreciate in the LAD region there are a few transcripts. Now we come back to our question. Why did we do this to begin with? We did this, we wanted to figure out what is the association of DSPs why lamin causes DSPs. Mm-hmm. Although this is not exclusive to lamin, but we use the lamin because lamin is known to regulate gene expression, because our data was hinting that transcription might be the stimulus for DSP. And we looked at here the distribution of double-stranded DNA break. Non-LAD region has more transcript and has more DSPs by about 6-fold. Interesting. Compared to another line of evidence that DSP may be linked to transcription. Time-wise, we have 5 minutes. Very good. Excellent. 8 minutes. So let me give you a little bit of role of transcription in generation of double-stranded DNA break. For transcription to happen, the blue is my double-stranded DNA, the dark blue. In order transcription happen, a bubble forms, the transcription bubble forms. The two strands get separated. RNA polymerase comes to binds to one strand, it starts making nascent transcript, and it moves forward, induces torsion in the DNA because it has to unwind the DNA. That torsion leads to those coils, the supercoils. That supercoils, they're both in backward and as well as forward, positive and negative. Those supercoils are become tight, would prevent RNA transcription to proceed. Something has to release it. And top two topoisomerases are assigned to this task, to cut this strand of the strands of DNA, release the torsion so transcription can proceed. As soon as they proceed, repair enzymes come, repair the site of double-stranded break. So that is the theory basis transcription and double-stranded break. So I will move on here and say the same thing that we should not ask. So what? What are the biological significance of double-stranded DNA break? I will not go into the transcription-mediated double-stranded break, which currently we're working on to delineate the mechanism. But I will move the translational aspect of it the last few minutes to have some meaning for those who may not be tuned to basic science aspect of it and more interested in translational part of it. To do that, what is the significance of double-stranded DNA break in cardiomyopathies? We have tested this in 2 different mouse models. To test this, we have blocked both the nuclear component of double-stranded DNA break by deleting T53, as well as the cytosolic component of double-stranded DNA break, which is mediated largely through cGAS. So in the next few slides, I will show you the data in cGAS. This slide, the Western blot shown there are 5 genotypes. The first 3 genotypes are the control groups. The 4th genotype is a mouse model of cardiomyopathy showing expression of showing expression of cGAS on the very top panel. you can appreciate compared to control, C-gas expression level is significantly increased in this cardiac myocyte, in this mouse model of cardiomyopathy, which is desmoplakin deficiency mice. We have done the same for lamin. Actually, I can't see whether this is lamin or desmoplakin. This seems to be lamin, actually. We have done both for lamin and desmoplakin. And the last genotype is deletion of C-gas gene, which is MB21D1, in the background of lamin deficiency. The top panel shows that you can appreciate that C-gas is largely gone, barely detectable. The downstream Western blot simply show the downstream cascade of C-gas pathway. None of them is completely abolished. They are attenuated because there are alternative pathways that activate them. Now we have done that. The most important phenotype to look for us is life and death endpoint, because we are, although in science we are biased and we may fudge the data, life and death is hard to manipulate. And here is red is the lamin deficient mice that you can appreciate by day 24, almost all of them are dead. And the green is effect of deletion of C-gas in lamin deficient mice. They extend the survival to up to around 35 days. Of course, one would not expect it to survive normally because the underpinning mechanism is still lamin deficiency. There are multiple other mechanisms involved, but nonetheless, this effect as good as the effect of beta blocker in heart failure, as well as the effect of ACE inhibitors in heart failure. So survival improved. We have done similar thing. This is the desmoplakin deficient mice. The first panel again, the three genotypes are controlled. The fourth one is desmoplakin, C-gas is increased, and the fourth one is deletion of the C-gas in desmoplakin. The blue or purple color is desmoplakin mice. You can appreciate most of them die within a month or two. And the yellow, dark yellow color is deficiency of C-gas in the desmoplakin mice, that these mice survive much longer and better. And the rest of them quantitative data. This slide shows effect on heart function. Again, the first three columns are control. The red one is lamin deficient, heart diastolic diameter is increased, deletion of C-gas attenuates that. The next panel is end systolic diameter. Again, the first three panels are controls. The next one is lamin deficient, end systolic diameter is also reduced. And so is ejection fraction, fractional shortening, and others. Effect on fibrosis. The first three panels are controlled. The fourth one is fibrosis in lamin deficient mice, which is increased significantly. And when you delete C-gas, it gets attenuated, which is the last panel. And the bottom is quantitative data. The next one is apoptosis by tunnel assay. We also have looked at the number of cell death pathway. We call it panapoptosis. And comprised of about a dozen molecules we look. Again, apoptosis gets attenuated upon deletion of C-gas in lamin deficient as well as in desmoplakin deficient mice. To conclude the last minute, we show that DSPs are increased, and DSPs are pathogenic because not only they activate the checkpoint TP53 cell cycle pathway, but they also activate the cytosolic DNA sensing protein pathway, leading to activation of NF-kappa B and IRF3 and inflammation, cell death, senescence, and cytokines. So if we block any of these pathways, I didn't show data on TP53, you can attenuate the phenotype. You can't cure the disease, you can attenuate the phenotype. That's where we are here at the present time, and we are moving to the next step, but it's a long journey. So I will conclude with one of my favorite leaders in the world, visionary leader, Winston Churchill. So every step may be fruitful, yet there will stretch out you an everlasting, ever ascending, ever improving path. You know you will never get to the end of the journey, which is true for us. But this, so far from discouraging, only adds to the joy and glory of the climb. So we are at the beginning of this endless journey. We still don't know, we really don't know much about this yet, and hope before end of my life I know something about it. Thank you again. Thank you very much for the opportunity to present
today. Great job, AJ.
Absolutely.
Wonderful. I love to end on that quote, but I think we have time for 2 quick questions.
Okay.
I'm going to ask one. So, double-strand breaks occur all the time. And are commonly occurring with transcription. That's what you showed us. It's accelerated in lamin-deficient animals and the progeroid patients. But it's happening all the time. And you said that what you showed us today had more relevance to everything besides cardiomyopathy.
Sure.
Tell us what you meant by that.
I think, to live, you have to transcribe. To live, you have to metabolize. Metabolites affect the DNA. Affected DNA lesions affect transcription. Affecting transcription increases DSP. DSP increases the pathway we discussed from cell senescence, cell death, apoptosis, you name it. So, therefore, aging is unavoidable. Cancer is unavoidable if you're long enough, because this genomic instability is constantly occurring. And single gene disorders have nothing to do directly with DSP. DSPs are secondary phenotype in single gene disorders, but contributes to the phenotype as I showed it to you. So there's no process in life in which transcription is not necessary. Transcription is essential for life, and consequently is DSPs. Of course, the second component of is replication. We didn't get into that myocyte by and large are non-replicating cells. But replication is the essence of double-stranded DNA break.
Yeah.
So there's no unavoidable.
So I guess my question is the part that you kind of skipped. So I'm interested in the basic mechanism why this lamin A as an envelope protein and its mutation or its disorder lead to the DNA leakage out of the nuclear.
Right.
What's the
That's right.
mechanism?
So there are one of the things lamin do, I call lamin also a guardian of the genome like T53. When lamin mutated, LADs get exposed, and those LADs become subject to transcription, and therefore DSPs occur at the site of those transcription that occur. But in addition to that, lamin probably helps to compact the chromatin, and when lamin is not there, the chromatin is less compact and more amenable to damage and oxidation or methylation, or indeed I'll show you the methylation effect of it, CpG methylation, which by definition is mitogenic, converting C to T. And so lamin is through various mechanisms, whether it's through effect on epigenetic chromatin compacting or DNA methylation, or simply transcription, contributes to DSP. A lot of them we have not directly tested, a lot of them based on literature that is already published. So our data, we haven't gone that far to detect why lamin deficiency. Our DNA is more oxidized at the LAD regions, we haven't addressed that. But we know CpG methylation increased. I'll show you the data. But there are more, again, this is the beginning of the journey. There are many unknown and answerable questions at the present time. Thank you.
Well, I'm afraid we're going to have to close there, but we'd like to welcome you back at some point to hear more about the journey.
Thank you.
You're at the cutting edge of cardiovascular sciences today. Thank you for joining us. Our guest today was Dr. A.J. Marian, talking about double-strand breaks and how they play an important role in cardiomyopathy.
Thank you both very much.
Thank you.